All right, the immune system has a number of receptors. In fact, what you'll find a lot of throughout this book is you're going to be talking about receptors and anti-receptors uh, that are used to recognize something. And then you're going to get a signal transduction pathway, which we'll talk about later in this chapter. Um, and the signal transduction pathway initiates some sort of response. So, a lot of the receptors of the innate immune system include things like the complement cascade. They're going to bind to negatively charged particles on the target microbial cells, and then they'll cause uh, opsonization and chemoattraction and all sorts of activities that can occur there. You also have mannose binding lectins. Since mammalian cells typically don't use mannose, especially in polymerized forms. We have an occasional mannose sugar found in a exopolysaccharide of some sort, but for the most part, we won't have a lot of the mannose containing microbial components. And so these receptors will bind to those, whether they've seen them before or not. C reactive protein is really of interest because it binds to phosphatidylcholine and pneumococcal polysaccharides that are found. So if you have good response with the C reactive protein, you're much more resistant to strep infections than the average person. Some people don't produce a very strong response and they get repeated infections with strep throat, not throats. Then, of course, there are anything that is unique to the bacteria, then you can use that as a way of recognizing and binding to it. So if you've had my microbiology class, then you realize that LPS is something that's unique. Peptidoglycan is unique. Some of the unique proteins that are found and cell wall components can be used as recognition receptors or recognition sites for receptors to bind to. And then the immune system can know that, hey, this is foreign because I don't produce it. One of my favorite receptors to talk about are these toll-like receptors, and they look like a shepherd's hook to me. The shepherd's hook reaches out and grabs the guy on stage and drags him off if he's doing a real bad job. Well, that's what these toll-like receptors look like. They're leucine-rich repeats that have very specific binding domains in them. And if they bind to something, let's say that this is binding to peptidoglycan. If this were to bind to peptidoglycan, then it sends a signal into the cell and lets the inside of the cell know that this is something that is not supposed to be here. So we need to increase phagocytosis. We need to start trying to bind this thing to us so that we can stop it from being able to cause infection. Now, there are many different types, and some of them will bind to specific gene sequences or lipoteichoic acids, LPS lipopolysaccharides, all of those different things. And so you're born with these. And when you're born with a specific type of toll-like receptor, then you have a good innate immune response to that particular thing. And you can see the different classes of these that are out there that can be even able to bind to double-stranded RNA, single-stranded RNA, DNA elements of bacterial cells. These are really important in the innate immune